Assalamu alaikum dear students. Today I will discuss Cloverleaf model of transfer RNA with the help of its diagram. Why do we call it cloverleaf? Because it resembles the clover leaf, which is a herb, and this leaf that has three lobes. As transfer RNA in its structure, it has three lobes like this clover leaf. That's why we call it what clover leaf model of transfer RNA. Now let's start it. This is the first lobe, then this is the second lobe, and this is the third lobe. Now I will draw the outline of it. So student, this is the 3' side of the transfer RNA and this is 5' of the transfer RNA. Like any RNA molecule, it is also single-stranded, but at times or at some places it's two strands or it's single strand that forms hydrogen bonds between the sides which are parallel to each other, but these are temporary hydrogen bonds and it appears like double-stranded, but actually it is not a double-stranded molecule, it is a single-stranded molecule. Student, at the 3' amino acid, it attaches itself. So this is the amino acid carrying site, and this 3' that always ends with nucleotides C, C and A. Always it ends with CCA, and this 5' that ends with a nucleotide, that is what guanine nucleotide. Now this loop is called as what? D loop. This is that very loop which attaches an enzyme with it, which can activate this transfer RNA for the attachment of what? Amino acid. That enzyme is called as amino acyl transfer RNA synthetase, which helps this transfer RNA to be activated for the attachment of what? Amino acid. This loop is called as what? Theta loop. This loop recognizes the position of this transfer RNA to bind with what? With ribosomes. Then this site is the third loop. This is called as what? Anticodon site. It is having 7 at this site, 7 bases they are attached, 7 nitrogenous bases they are attached, or 7 nucleotides they are there, and the middle 3 they make an anticodon. And always this codon which is present on the transfer RNA, that is anti to the codon present on the exposed strand of the or exposed codon of the messenger RNA. That's why it is called as what? Anticodon. For example, if on the messenger RNA, this is a messenger RNA strand, 5' and 3'. So like if it is having guanine, adenine and uracil, so on the transfer RNA, anticodon must be having 3 nucleotides, like opposite to guanine would be what? Cytosine. Opposite to adenine there will be uracil, and opposite to the uracil there will be what? Adenine. So this kind of combination that will be there on the anticodon. Okay student, transfer RNA that consists of about 75 to 90 nucleotides, and this transfer RNA that is considered as the smallest RNA molecule, and it makes 10% of the total RNA of total RNA. Like its percentage is about what? 10% of total RNA. So that's all about transfer RNA. Transfer RNA function is to carry particular amino acid to the ribosome, where messenger RNA that is attached for the synthesis of what? Proteins. And for every particular kind of amino acid, a particular kind of transfer RNA that is present in the cell. Thank you students.